Yeah, welcome everyone. So excited for your interest in this wonderful group of humans that you see on the screen here. So just want to give you a little bit of background about what the Global Change Research Fellows program is about, so you can know what these folks are currently doing and how other folks might get involved down the road. So Myra, if you could scroll on to the next one. Next slide, if you will. Yeah, so we at NC State are the host institution for a consortium of institutions across the Southeast. These are our consortium groups, and the fellows are all from one of these consortium institutions. So if on the next slide, you'll see who is eligible to become a Global Change Research Fellow. The students need to be at either one of our consortium universities or a citizen of a USET, the United South and Eastern Tribes member organization, a tribal nation involved in that organization. So as part of the Global Change Research Fellows Program, we provide a year of research assistantship support, plus tuition and health insurance. So we fund a small portion of these folks' overall research programs in either a master's or a PhD program. So next, you'll see that what these fellows are about. So they are doing their own research, and that research is part of the mission of the larger Southeast CASC. So falls within the overall research portfolio of what the Southeast CASC does. And they're an interdisciplinary group, which you'll hear about shortly. And we just lost that. In any case, so they come from a lot, and they are advised by someone in their institution who is a research affiliate with us. So next, you'll see what they do, what they're required to do as part. They do their own research. They also are involved in a team science synthesis project. We have a great field intensive as a kickoff for their research support for the course of the year. And they coordinate some public-facing seminars. So look for those announcements coming down the pike. And we do some professional development, skill building workshops over the course of their year. Yeah, and just real quickly, this gives you an example of some of the things they're involved in. So they're coming out of here pretty well-rounded and ready to do some great co-produced science. So commend them into their next steps of their career to you. So I think that's it. Yeah. So the timeline, we will be recruiting for the next cohort of fellows, and this is our timeline for how that process will go. So we're going to try to get that call out late this month, take applications, and then do a review of those and selections, hopefully to get some folks awarded in mid-April, so they can make those decisions, and then their term will start August 1. So now I'll turn it over to this great group of folks, and yeah, take it away, students.
Thanks, Carrie. And if anybody has any questions about the Fellows Program, please, again, feel free to use that Q&A box, and Carrie can answer your questions as the presentations are going. Without any further ado, Asumi, please take it away.
Great. Hey, thank you for introduction. Hello, everyone. My name is Asumi Saito, first-year PhD student at Center for Geospatial Analytics. Do you know your home is safe from flood? After witnessing the devastating damage that Hurricane Helene brought, this is the best time for you to check your safety and prepare for future floods using a flat map. A flat map is a map that shows floodplains that are susceptible to certain levels of floods, such as once in 100 years level of flood. However, the maps are often created based on the stream gauges, and the areas far from the gauges, such as urban areas, would not have precise prediction. Also, the map only updates once in 5 to 10 years, which means rapidly developing areas would not have accurate information on the flood maps. With accelerating climate change, we are experiencing have more frequently extreme floods, and Therefore, the accurate and timely forecasting of the event is more important than ever for regions at risk of disasters and their vacation planning. My research uses a method called height above nearest drainage, HAND, to predict flood inundation by incorporating the elevation gap between the areas close to the stream and far from the stream gauges. The HAND method uses past stream data, elevation gaps, and supplementary information about inundation, such as city drainage information, to create a near-time inundation map. The GIF here shows the change in the inundation area during August 2018 when Hurricane Michael hit the Tall River Basin, Rocky Mount. The photo in the background is the city of Rocky Mount after the hurricane. You can see the whole town is inundated. This is preliminary work, but you can see the day-to-day change here, and the change is rapid. Using the HAND method, we can feed near-time stream information to create an emergency warning to flood-affected communities. For example, what if the flood level goes beyond what a flood map expected? The near-time inundation map updates the warning as soon as it receives the stream data. Also, the hydrologic reasoning behind the method provides explainability to the decision makers for a swift response. In my future work, I will add satellite data to develop a method to create an inundation map that is accurate, near time, and explainable. A flood map serves for safety only when it is well utilized. Therefore, along with model development, I want to make the map easy to access for communities and easy to utilize for planning preparation and vacation. Thank you.
Thank you so much, Asumi. Appreciate it. Folks, if you have any questions about Asumi's mapping project, please feel free to put them in the chat. But now we're going to move along to Chris. Chris, feel free to share your screen and be ready to present.
Thank you. So hey everyone, my name is Chris Oates. I am a second-year PhD student in biological and agricultural engineering at NC State, and my work is centered around social vulnerability and nutrient water quality monitoring. So currently in the United States, a lot of federal water quality monitoring networks do not currently consider community demographics when establishing their monitoring systems, which means that communities that might be more socially vulnerable, lower income, less affluent communities, that's not something that's that is brought into consideration when establishing these stations. And that means that states or communities that are more socially vulnerable, we know that those communities are at risk of, suffering more environmental racism and different environmental injustices. But it also means that these communities don't have access to environmental data and environmental monitoring data that can help them quantify and actually advocate for themselves when it comes to water quality monitoring issues. So in my research, we've been using data from the EPA's water quality portal, and that portal synthesizes a bunch of data from the USGS, the EPA, USDA, and a lot of other state and local agencies as well. And it puts all that data into one easily accessible database that has all the water quality monitoring information for the United States. And with this data, we've been able to look at where these stations are placed. And what we've found through preliminary research is that water quality monitoring stations that look for nutrients are heavily biased towards being placed in census tracts that are less socially vulnerable as opposed to more socially vulnerable. In the bottom left-hand corner of the screen, you can see a heat map of where the nutrient monitoring stations are located relative to the American Southwest or Southeast. Moving aside from Florida, which does a pretty good job of monitoring pretty much everywhere, we see there's a heavy concentration of stations around metropolitan areas. There's a lack of stations in some of the more rural and more socially vulnerable areas. The map in the middle shows social vulnerability. The colors that are more red are more vulnerable. The bluer colors are less vulnerable. So if you look at those two maps, you can see that the stations are really located towards cities and areas that are more affluent. And what this work can mean for the future is that essentially we would like for regulators and environmental policy to factor in vulnerability when creating these stations. A lot of areas that we know suffer from lower water quality, poorer water quality, are not being monitored at the rate that they should be. Our analyses have shown that the rates of monitoring are drastically lower than they should be. And this also has a large societal benefit as well. We know that communities that are more marginalized and underrepresented and under-resourced don't always have the ability to advocate for themselves on the environmental level, and adding more monitoring that is a lot more holistic and looks at community needs as well as, you know, agent feasibility and things of that nature would really help out a lot for these communities. In future work, we know this is a problem. This work has already been written up and is under review currently. But future steps will be to actually assess water quality in stations or in census tracts that are more vulnerable to see, you know, just how drastic these water quality outcomes are. And then the next stage after that is actually developing machine learning models and whatnot that can actually help predict water quality in lieu of the stations not being there. So thank you guys.
Thank you so much, Chris. And again, I'll encourage folks who are watching, please put your questions in the chat. Even after people's talks, if you want to just flag who the question is for, they are waiting to reply to your questions. Next up, Gwen, if you want to go ahead and start sharing your screen.
Yeah, hello. Yeah, so my name is Gwen Kirschke. I'm a second-year PhD student at the Center for Geospatial Analytics at NC State. No. And my background is in field ecology, and I've done a number of kind of projects in that vein. One looking at monarch butterfly populations on native and non-native milkweed host plants in northern Florida, which is what I'm doing there on the right. And then I've also spent the last several summers out at the Rocky Mountain Biological Lab in Colorado, looking at the impacts of drought on floral abundance and in particular in the quantity and quality of floral nectar that's produced as potential resources for pollinators like bees. And so now, as a grad student in geospatial analytics, I'm looking at methods that can scale the types of information that we collect in the field, kind of at specific points up to continuous maps. And in particular, the project that I'm working on right now involves taking the floral nectar data that I had collected and combining that with high resolution hyperspectral aerial imagery from the NSF NEON program, and using that to create continuous maps of kind of the density of floral nectar that's available throughout the meadows in this system. And while I'm doing this work in Colorado, because of the availability of the field and imagery data that I need to work on that project. Part of the point of the project is to try to test out methods that would be replicable in other systems, including in the Southeast. And having these types of maps allows us to understand where there are the resources that can support pollinators, such as bees, and also how the distribution of those resources is potentially going to change with things like climate change and changes in land use, which can allow us to assess kind of management priorities and where we might want to put resources in terms of supporting those insect populations. Thank you.
Awesome work. Thanks very much, Gwen. All right, next up we have Hannah. Hannah, feel free.
Hi, everybody. Oh gosh.
Hey, you might have to just hit presentation because we can see your whole screen.
Okay.
There we go. Perfect.
Okay,
Oh,
great.
nope,
So
now we lost
hi,
everything. Sorry, Hannah, we lost it all. Now we
Oh.
have your R screen, which is just, there we go. Okay, perfect.
Oh my goodness. Okay,
There we are.
So hi, everybody. My name is Hannah Ferraro. I am a current master's student at the University of the Virgin Islands. I am in the Marine and Environmental Science program. So I am currently working on developing my research questions. However, I am
Sorry, Hannah, just one quick one. We can see your full screen.
Oh, Oh, no. I'm so sorry.
No worries.
Is this okay? I think now I might have that.
Perfect. We see your other slides, but this looks great too.
Okay.
Perfect.
Okay, great. So like I said, I am currently developing my research questions. However, I am very interested in seagrass systems, and I intend to pursue blue carbon storage as my thesis work. So seagrass meadows are one of the most powerful ecosystems on the planet when it comes to carbon storage. Known as blue carbon ecosystems, they sequester large amounts of carbon in their biomass and sediment beneath them, playing a critical role in climate mitigation. But what exactly happens to their carbon storage ability when these ecosystems are disturbed? So this is the question that I aim to explore in my research. My focus is on comparing how different species of seagrass handle carbon storage after environmental disturbances, such as storms, coastal development, or pollution. In particular, I'm interested in understanding how resilient these meadows are and which species are the most efficient at continuing to sequester or store carbon after such events. As many of you maybe know, seagrass species differ wildly in their morphology, growth rates, and environmental tolerances. Some species like Zostera marina, otherwise known as eelgrass, which is found in more temperate regions, have evolved to withstand cooler, more nutrient-rich waters, while tropical species like Thalassia testidium, also known as turtlegrass, thrives in warmer, more clear environments. These differences could suggest that certain species are better equipped to recover from disturbances and continue to store carbon better than others. I'm going to be conducting my thesis work in the Caribbean, either off of St. Thomas or St. John. So the 4 seagrass species that I'm interested in studying, going from the top left and working clockwise, is shoalgrass, manatee grass, turtle grass, and then holophila, which is an invasive grass influencing the area. So why is this important? Well, post-disturbance resilience directly affects the long-term viability of seagrass meadows as carbon sinks. If some species are more efficient at storing carbon after a storm or human impacts, they can become key targets for restoration efforts. For example, in areas experiencing more frequent hurricanes, a species maintaining a higher carbon storage, despite regular disruptions, may offer a more sustainable solution for blue carbon conservation. Ultimately, my goal is to help inform conservation and restoration strategies by identifying the species and conditions that support the most effective and long-lasting carbon sequestration, and which could be vital in regions where climate change impacts like stronger storms, rising sea levels, are a more constant threat to these coastal ecosystems. By understanding which species of seagrass provides the greatest resilience in the face of these disturbances, we can prioritize those specific species in our restoration efforts, which will ensure that blue carbon storage will continue to play a critical role in climate adaptation and mitigation. Thank you.
Thanks so much, Hannah. Appreciate it. All right, next up, Bigyapti. Bigyapti, feel free.
Can you see the screen?
Perfect.
Hello everyone, I am Bigyapti Nepal, a first-year PhD student at Center for Geospatial Analytics at North Carolina State University. Today, I'd like to talk about a serious environmental challenge, harmful algal bloom or HABs. So these occur when algae grow out of control in water bodies, often due to excess nutrients from agriculture, urban runoffs, and many other causes. And these blooms produce toxins called cyanotoxin that can poison aquatic life, as you can see, a fish dying because of that, and makes water unsafe for humans, leading to fish kills and health risks from drinking or eating the contaminated water or food. What if we could predict? What if we could predict when and where these harmful blooms might occur? So we could act before they can cause any damage. So my research aims to predict when and where HABs will occur, or also looking at the pattern where they've occur by looking at variables and using a method called Bayesian network, which brings together different data sets from different variables like water temperature and nutrient levels, salinity, and also using remote sensing to detect it. And and by this I look forward to developing a predictive model. This allows us to map areas at a risk of harmful algal blooms before they cause any damage. So what makes this approach different is that we're using the principle of co-production to work with and by working with partners like local communities and environmental agencies to ensure that the tools that is being developed are practical and useful in the real world. So now why HABs matters. So this is important to all of us across different departments and disciplines. And the problem of HABs is not just an issue for environmental scientists. It affects public health, agriculture, economy, and even social equity. So clean and safe water is a shared resource, and the better we understand and predict threats to our water systems, the better equipped we are to protect it for everybody. So in closing, I believe the fight against harmful algal blooms require collaboration, and while my research focuses on creating predictive tools, their success depends on working together across fields from science, policy to local communities, and together we can help protect our water and ensure safety for all. Thank you.
Wonderful. Thank you so much, Bigyapti. And again, folks, we are waiting for your questions in that Q&A box, so feel free. Next up, Allie.
One second, sorry, I'm trying to figure out are you guys seeing my screen?
Yep, we can see you and we can hear you.
That's okay.
Yep, looks great.
Hey everyone, my name is Allie Brown. I am a doctoral researcher at Auburn University. I wanted to introduce you guys to my field of study, which is called discipline-based education research, otherwise known as DBER. And within this, discipline-based education research combines geoscience content or disciplinary specific content with complementary research in social science and human learning in order to advance teaching and learning in a given discipline. I specifically study geoscience education research within this umbrella in order to study how people think and learn about climate change here in the Southeast United States. And like I said, my research is focused in the Southeast United States, which according to the most recent National Climate Assessment is home to the most frequent and expensive climate weather-related disasters compared to any other region in the United States. Despite this, however, people in the Southeast United States have the lowest comparative climate literacy rates when compared to the overall national average due to a number of complex socio and political factors. But we want to understand how we can best reach people in the Southeast United States and teach them about climate change effectively. And so my research utilizes a pre-post study design in order to assess student understanding and perceptions at the beginning and end of the semester using a validated survey. So we use this pre-post design in an introductory weather and climate class that targets non-science majors in order to teach them about climate change. And so this survey consisted of around 20 content-related questions, 7 perceptions and beliefs questions, and a few demographic questions like gender identity, major, and home zip code. Really, the ultimate goal of this study is to improve student knowledge and perceptions of climate change. We can understand how students are learning, what students are learning. That way we can modify this class in the future and better reach a wide array of learners. And so from these results, or from this data collection that's been taking place for the last 4 years, we had 164 paired responses. And some of these preliminary results show that content knowledge, which is the box plot on the left, belief in climate change, the box plot in the middle, and risk perception of climate change on the right, all show significant increases in student beliefs or attitudes towards this topic. So for content knowledge, for example, the average increased by about 5 to 7%. The belief in climate change increased quite dramatically. The scale is reversed here, so the lower number is more sure of climate change. And lastly, risk perception of climate change also increased statistically significantly. And so next in this data, we want to conduct some internal reliability and make sure our survey performed as expected. And then also we want to dig into this data a little bit more so that we can determine some indicators of significance, such as major, gender identity, or home zip code, if those are predictors of the results we're seeing here. And this is so important because climate change affects everybody. And so reaching these non-science majors that will be our future decision makers is incredibly important to make sure that we are better equipped to mitigate the challenges that climate change will inevitably pose. Thank you.
Ellie, thank you so much. Wonderful. Jamie, feel free to take control of the screen. You're up next. And we can see you, but we can't hear you yet, Jamie.
All right, so you can see my slides right now?
Yep, perfect.
Perfect. Picture this, you spent Sorry about that, the video.
No worries.
Picture this, you spent months planning and saving for a family getaway to the beautiful Choi River in eastern North Carolina. You finally arrive, the kids are excited, swimsuits are on, but then you see the sign. The river is closed due to an algal bloom. All that anticipation, all that hard-earned money, and now the water is off limits. This isn't just a ruined vacation. It's a glimpse into the environmental challenges we're facing today. Hello, everyone. I'm Jamie Huerta, a master's student in Geospatial Information Science and Technology at North Carolina State University. My research focuses on the Albemarle Sound, an important estuarine system in North Carolina that has been increasingly impacted by harmful algal blooms, specifically cyanobacterial blooms. These blooms not only ruin vacations, but also pose serious risks to both human and ecological health. So what's driving these blooms? The answer lies in nutrient pollution, particularly nitrogen. Nitrogen is a key ingredient for algal growth, and when there's too much of it in the water, it leads to the rapid, harmful growth of algae. My project, which spans from May of 2022 to September of 2024, aims to understand how nitrogen, especially in its organic form, fuels these blooms. While inorganic forms of nitrogen, like nitrate or ammonium, are easier to track, the organic forms are more complex and require advanced techniques to analyze. That's where my research comes in. I'm using a statistical model called FluorMod version 4, which helps identify discrete sources of dissolved organic nitrogen based on their optical properties. Essentially, the model allows us to track nitrogen back to its sources, whether it's runoff from agricultural fields, urban areas, or natural environments. With the help of monthly data collected from 13 monitoring sites from 4 different watersheds across the Albemarle Sound, I'm studying how nitrogen inputs change over time and space. This information, combined with land use data from the National Land Cover Database, allows me to investigate how activities like farming, urban development, or deforestation are contributing to nutrient pollution. The broader goal of this research is to provide resource managers and policymakers with the tools they need to target specific sources of nutrient pollution. By better understanding where nitrogen is coming from and how it's affecting water quality, we can develop more effective strategies to reduce nutrient inputs and ultimately prevent harmful algal blooms from even occurring. That ruined vacation, it's much more than just a personal inconvenience. It's a wake-up call for the environmental challenges affecting our waters. By taking action based on research like mine, we can work to protect places like the Choi River for future generations, ensuring that we can continue to enjoy these important ecosystems, both for recreation and for the health of our environment. Thank you.
Wonderful. Thank you so much, Jamie. All right, Henley, feel free to take control of the screen.
Hi, everyone. I'm Henley. I am a master's student at the University of Tennessee, Knoxville in ecology and evolutionary biology, and I'm interested in conservation planning. So the overall goal of conservation is generally to preserve biodiversity and natural resources. And a common strategy is to establish protected areas to support species, especially since we know that habitat loss is a major driver for species decline. And so conservation actors can make strategic plans on where to invest and the most optimal places to establish these protected areas in order to achieve these conservation goals. We know that planning for future conservation efforts has to take into account climate change because climate change is affecting where species can inhabit. And so continual evaluation of our conservation planning is going to be helpful for making sure that these efforts are in the right location. So my overall question is, how effective are collaborative conservation planning maps at capturing areas that are climatically suitable for species under different climate scenarios in the future? And so a great example of some collaborative conservation work is through the Southeast Conservation Adaptation Strategy, which has made this map called the Blueprint that is a great map of the Southeast that identifies areas from highest to low priority for conservation efforts. And this is collaborative with more than 600 organizations involved, over 2,000 people, and they are using natural and cultural indicators to determine the optimal location for highest priority, for example. And so I'm comparing this map against ecological niche model data for 322 species, specifically in the Southern Appalachian region under a host of different climate scenarios. And I'm looking at the comparison here. So I'm performing linear regressions to compare the area that's climatically suitable for each species within the extent to the area climatically suitable within the priority. So if we assume a null hypothesis of random selection of priority areas, we would expect to see a one-to-one line as the species that have areas suitable in the extent should be proportional to the area that's suitable in the priority. Early results, so for example, here looking at specifically birds under a full dispersal scenario for one of the climate scenarios, we can see that the highest priority is performing better than this one-to-one line with a slope that is less than 1 and a positive intercept. So this suggests that the rarer species that have suitable area in the extent are being represented more in the highest priority area, which is a good thing. So the next steps for me are to keep looking across scenarios, incorporating measures of dispersal and looking for any trends. Thank you so much.
Great. Thank you so much, Henley. Okay, Grace, feel free to take it away.
Okay, can you see my slide?
Yes, we sure can.
Okay, great. Hello, my name is Grace Choi, and I'm a third-year student at NC State University in the Center for Geospatial Analytics. I'm currently part of the Spatial Ecosystems Analytics Lab, which is led by Dr. Josh Gray. My current work centers on vegetation phenology, which you can think of as nature's calendar, tracking the life cycle of plants throughout the year. Just like our year has a beginning and an end, plants follow a similar rhythm from the start of the growing season to its conclusion. My focus is on the end of the growing season, a critical period that's becoming harder to predict with climate change. In northern ecosystems, research shows that growing seasons are lengthening mostly due to rising temperatures that affect the start of the growing season. This could mean more carbon is absorbed by vegetation over a longer period. However, what happens at the end of the season is still unclear. Some studies suggest it's ending earlier, others points to delays, and some show no change at all. So understanding these shifts is vital because they impact global carbon, water, and nutrition cycles. And plants absorb carbon during photosynthesis and release water vapor through transpiration, processes that are essential for regulating climate and keeping our ecosystems healthy. My current work focus is on drylands, which covers 40% of Earth's surface. and sequesters 30% of the world's carbon. Despite their importance, there's a lot we don't know about how the end of growing season in these ecosystems responds to climate variability. The end of the growing season is especially sensitive to resource limitations like temperature or water or nutrition, which vary across different regions. So I'm exploring how these resource limitations affect grasslands, particularly in the US and Africa, where there's more sensitive to water shortages and other factors like temperature. And so my work focuses on how those sensitivities shape the end of the growing season, especially during times of droughts and floods, hoping that understanding these patterns can give us better insight into how climate change might impact some of the planet's most crucial landscapes. Thank you.
Thanks so much, Grace. Okay, next up, Rachel. Rachel, take it away.
All right, thank you. You can hear me okay?
Can hear you okay? Just don't see any slides yet.
Sharing now. All right, how's that look?
Perfect.
Awesome. So hi, my name is Rachel. I'm at NC State in the Hartstone Rose Comparative Anatomy and Functional Morphology Research Lab. And today I'm going to be talking about man-made evolution. So in today's day and age, change is rapid. Humans are always working to innovate everything around them. And so other organisms are constantly working to keep up with these changes. So as a fellow of the Southeast Climate Adaptation Science Center, or CCAS, I have the unique opportunity to study how human-driven changes to the environment are influencing animal morphology. Humans have long influenced the physical forms of animals through activities such as climate change, through humidity and temperature, forced migration, urbanization, captivity, domestication, pollution, and so many more. However, in recent decades, the scope of our impact has grown. All these factors have caused shifts within environments and available resources that forces these animals to adapt. These anthropogenic effects are altering the very anatomy and morphology of these species in ways we're only beginning to fully understand. So we don't really consider what we're doing when we leave our porch lights on at night, for example. So one lesser known influence is artificial light at night, or what I call ALAN. So my research focuses on how ALAN is driving these rapid morphological changes, particularly in Gambusia holbrooki, which you might know as the eastern mosquito fish. Artificial light is just one of the many byproducts of human expansion, and it has far-reaching impacts on wildlife that we wouldn't really consider. In the case of the eastern mosquito fish, constant exposure to ALAN is causing significant changes in their body structure. So these fish are living in populations within ponds near these large buildings that have constant straight lights on at night. And it's changing their anatomy significantly and evolutionarily. So most notably, what we're seeing is the caudal fin, which typically has a shovel-like shape, which is suited for slower, more deliberate movements. It's evolving into a triangular shape, similar to that of a tuna. So A would be the more rounded shape, and it's becoming more truncated, like B there. This truncated shape is associated with more speed and agility, whereas the round shape is associated with small bursts of speed, not a constant stream. This shift results in that more streamlined body and more streamlined athletic body, which appears to be an adaptation that we're seeing caused by this artificial light at night, as they're starting to continuously swim throughout the day and the night instead of the small bursts of energy. So this research is important because it kind of highlights how seemingly minor aspects of human development, like streetlights and urban glow, can trigger profound evolutionary changes. By understanding these changes, we can better predict ecological consequences and explore ways to minimize the unintended effects of our actions on the natural world. Ultimately, the study of human influence changes to animal morphology is actually vital for conserving biodiversity and ensuring that ecosystems can continue to function in a rapidly changing world. Thank you.
Great. Thank you so much, Rachel. Next up, and I'm gonna, oh, Maida is just on it. Thank you, Maida, for queuing up this slide. Next up would be Kurt, who's one of our fellows this year as well. Kurt unfortunately couldn't make it today, but Kurt provided me with a brief paragraph here to read on their behalf that describes their research. So I'll just suggest if you have any questions, we can throw Kurt's email in the chat as well, so that you can reach out directly to them. Okay, so this is from Kurt. Their research looks at relationships between communities and scientific institutions that are created to help solve complex problems related to climate change and environmental justice. Specifically, they're using participatory, action research qualitative methods, which is being conducted with sharing circles and collective memory work to understand and document Indigenous peoples' experiences in scientific collaborations, which they go on to define as deliberative, co-produced science and participatory science between Indigenous collectives, such as tribal nations or Indigenous-led organizations, and Western institutions, such as universities or government agencies. They're also using reflexive qualitative methods with non-Indigenous people who partner with Indigenous groups to understand their values and how those values are integrated into their organizational practices and personal approaches to creating relationships with their partners. Their research aims to understand how conflicts or synergies between Indigenous ways of knowing and Western scientific practices, historical context, and trust-making/repair, and ethical issues such as data sovereignty influence these collaborations. And they are emphasizing relational accountability and the 5 R's of Indigenous research, which include relationship, relevance, reflexivity, respect, and reciprocity. As a way to understand the culture and nature of scientific collaborations, and to help create context-specific frameworks for making empowering relationships rather than extractive partnerships. So that's a little bit about Kurt's research. And again, I encourage you, if you have questions for Kurt, I'll put that email address in the chat briefly, and then you'll be able to reach directly out to Kurt. Next up is a video. Halina is here with us, but Halina is stuck in a car. So what Halina has done is prepared a video that's exactly 3 minutes long. Way to go, Halina, that will describe their research. So Maida, take it away. And I can't hear the audio.
I was afraid of that. I'm not seeing, oops, not seeing an option to share audio. Oops.
I think maybe I heard it for a second there.
Good afternoon. My name is Halina Pyzdrowski.
Perfect.
I'm pursuing my master's of landscape architecture at NC State University. Normally, I would begin something like this by asking you to imagine that you've just wrapped up a neighborhood barbecue in your backyard. There were hot dogs, roasted corn on the cob, games. You just caught up with some friends that you hadn't seen in a while. And you fall into bed content and grateful for all the relationships that you've formed over the years. Then, in the middle of the night, you awake to a crashing sound. You look out the window, disoriented, and before you can even start to think what's going on, you see fast-flowing water where your backyard was just a matter of hours ago. Unfortunately, we don't have to imagine a scenario like this because we in the Southeast have just witnessed one of the most catastrophic flooding events ever. And as shocking as it is to see a devastation of a place like Asheville, many of the small communities in the mountains have literally been wiped off the map. And this isn't uncommon in climate disasters, right? It's often the most vulnerable communities. And I'm using vulnerable here as the capacity for an entity to respond to stressful events that are most adversely affected. And this is because we have a scale problem. Climate change and its impacts, as well as the agencies that we've designed to respond to them, are vastly larger than a single community. And so it's often really difficult for small communities to get the resources that they need, not only to recover from a large-scale flooding event, but also become more resilient to future events. And so this is the problem that I work on. Currently, I'm working with the Coastal Dynamics Design Lab at NC State and the Coharie Tribe in Sampson County, North Carolina. Besides being the homeland for the Coharie people, Sampson County is one of the most agriculturally rich and significant counties in the state. And so as a part of this larger effort, I'm plugging in at this agricultural flood control management phase of the project. Specifically, I'm using a GIS-based tool to cite the agricultural practices on the land, like constructive wetlands and riparian buffers, to help improve the water quality as well as flood resiliency measures. And hopefully, like this image on the right here, the Coastal Dynamics Design Lab put together before my involvement on the project, getting people to visualize how these practices might benefit the agricultural system as well as the community at large. So in conjunction with all of this and co-design sessions with the Coharie people, our work is really trying to build capacity for the tribe to become the facilitators of watershed management in the county while enhancing culturally significant fishing habitat and river access.
Wonderful job. And I'll just tell everybody who's attending, Halina is here. I believe she's able to answer any questions you might have in the Q&A. So feel free to put your questions in there. And last but certainly not least, taking us home this afternoon, Mileisha. Mileisha, feel free.
Yes, I'm going to share right now. I hope everyone could see my presentation. Can I get a compliment? Yes. Okay, so my name is Mileisha Velazquez. I'm currently in my second year as a PhD student. I decided to pursue this degree with a background in biology and marine sciences to become a contributor to marine pollution and find ways to improve our water quality and our ecosystem health. So this is a short 3-minute thesis presentation of what I'm proposing to do. So I titled this Intricate PFAS, as in per and polyfluoroalkyl substances, or forever chemicals, and their interactions and biological impacts to coastal ecosystem health. And if I were to summarize in a sentence what I'll be doing, it'll be the following. I track PFAS contamination in coastal ecosystems by researching its biological impact and in seagrasses and microbial communities so that we can improve ecosystem health to better inform mitigation and bioremediation strategies concerning these types of forever chemicals. The target species that I'll be researching are specifically manatee grass and the sediment and biofilm microbial communities. Biofilm just means that they have the ability to form this substance that makes them stick together better. So it's like a micro environment for them. But where specifically? We're here in the state of Florida in the Indian River Lagoon. And why the Indian River Lagoon? Well, the lab that I work here at UF with Dr. Catherine Delis-Quiñones, the lab has found a large diversity of PFAS mixtures and concentrations that are there in the Indian River Lagoon, which is linked to cancer in those coastal vulnerable communities that are constantly exposed to these PFAS mixtures. And so we know that there is a human health concern to these chemicals. There are thousands of them. Not all of the information is readily available since most of them is proprietary. So there is not much that we know about the impacts, but we do know that it is widespread and persistent. It has an ecotoxicological effect and a bioaccumulation potential. Here, I just made a quick figure try to summarize it, people who don't know what PFAS is, it's a forever chemical. I just put two of the main ones that are studied, but there's so much more that we need to know. And they come in strong carbon and fluorine bonds that make them so persistent. They're very strong and hard to degrade, so they persist in a very long time within the aquatic ecosystem, which is a primary sink of these pollutants. That's why it's a site of concern. And they are found in many applications and many products, our day-to-day cosmetics, or even fabrics, electronic devices. They're great doing their job, but their effects once they reach these ecosystems, specifically what I'm going to be talking about is the coast, well, they have detrimental effects that we yet don't have much information on. And one of the chapters I'll be including microplastic as another type of forever chemical. Microplastics have additives, and one of these additives are PFAS, and they could even absorb other types of PFAS. So it could get even more complicated as these coexist together and they interact with one another. So what does that mean to these aquatic biota, and hence us, really? So just to give a background on why is it important on seagrasses and why microbial communities, they're so small, they won't matter, but they serve as ecosystem engineers. So they facilitate other processes. For an example, the microbial communities engage in chemical communications to help other organisms to settle. The manatee grass helps as a substrate to hold down coastal erosion, for example, and very productive. So they also they create habitats, and they have these interactions with other species. So they are very important to maintain the ecosystem health and hence our health. So this is why these two are important, and they're actually not much understood. They're understudied, along with these PFAS mixtures that are understudied as well. So there's a lot to unravel here to know, to have better information and inform our management. So in a nutshell, it's a lot to unpack, but the objectives of my proposed research involve knowing more about how the PFAS specifically is bioavailable, how it behaves within the interface of sediment and water, how it absorbs, deabsorbs, and becomes bioavailable for biouptake, and characterize the communities of these microbes and hence their function, how their biogeochemical cycling that promotes ecosystem health and carbon, nitrogen, and phosphorus cycling, how is that impacted with these natural gradients of PFAS within the Indian River Lagoon. And lastly, coming back with the seagrasses, I aim to explore also their gene expression, how these change within these gradients as well, and to have more information upon that with the PFAS mixtures, not with just the two that are most commonly studied. So that'll be my proposed research in 3 minutes. And I like to finish off with this quote from Sylvia Earle: "Health to the oceans means health to us." And thank you very much for your time.